we go. Okay, sorry. So we completely forgot to record it, but yeah, you haven't missed much on the recording. We're still good, so we're still good. Okay. Okay, good. So going back to the topic of cellular agriculture, just as a reminder for those of you that are not very familiar or you've only recently joined the UCL Alternative Protein Society, the term cellular agriculture was first introduced in 2016 by Isha Dattar. She is the New Harvest founder. And basically, cellular agriculture refers to the production of animal-derived products using cell culture techniques and biotechnology. Are those comments coming in? Is that what I'm hearing? Ah, okay, I see. Okay, so in terms of examples of products that are captured within the cellular agriculture field, that includes anything that is derived from animals, and you'll see I've put up here a few things related to foods, but also to materials like textiles. So silk is derived from silkworms. Leather is also an animal-derived product because it's derived from the skin of animals. And I've also added vanillin. Even though vanillin is not an animal-derived product, there are actually quite significant ethical issues related to sourcing, particularly through the use of child labor work. And vanillin, so the actual vanilla flavor is given by this compound called vanillin, which can be produced through cell culture using cell culture techniques and biotechnology. So in a way, even if it is not derived from animals, it can still be produced and it can still have benefits. So in terms of the alternative proteins product, and this is actually slightly more extended. So while cellular agriculture focuses particularly on animal-derived products, alternative proteins is more like an umbrella term where it also encompasses plant-based proteins. So in the alternative proteins category, in the alternative proteins space, there are essentially four main categories of products. You can have plant-based, recombinant-based proteins, non-animal cell culture derived, and these are mainly focused on fungus and algae. And then you can have the animal cell culture derived products. And I'm sure a lot of you are familiar with at least two of these categories. So the plant-based proteins have reached the market. Those are commercially available. And actually, since a couple of years ago, they have gone better and better. And then non-animal cell culture products, which are based on fungus or microalgae, particularly the fungus ones, I'm sure some of you are familiar with, probably tried them, tasted them. And these are, particularly the Corn brand, which is a British brand that now has actually picked up quite a bit and now is internationally available. And especially in the US, it seems like it's a big hit as well. And then the other two categories of recombinant proteins, these are essentially things like milk or cheese or gelatin, so anything that is based on molecules such as proteins that can be produced using recombinant DNA technology. And then animal cell culture products, of course, you know, it's cultivated meats, and that also refers to cultivated fish products or crustaceans products fall within the same category. So the ones that the one that I will particularly focus on is the cultivated meat one, which obviously I've tried to link it to the major milestones that have recently taken place. And in terms of cultivated meat, because I think this will spike quite a few of the discussions, The concept of cultivated meat is that basically this is genuine animal meat, is just made in a different way, made using biotechnology and cell culture techniques, but theoretically will mimic animal-derived meat down to its molecular level. So it will have the same composition in terms of the cell types. It will have the same nutritional profile, the same taste, and actually because it is made in a controlled way, it could actually be better from a nutritional point of view, and it could be better for the health of people as well. So these are just a few examples of some of the cultivated meat world's first. And of course, I'm sure all of you are familiar with Mark Post's first cultivated beef burger that was showcased in 2013. But then there have been a lot more afterwards. And what I really wanted to show here is the variety of products that can be produced using this concept. So not just burgers, but you can also make meatballs, fish, cakes, etc., bacon, and also the species. So you can make beef, pork, fish, shrimp, etc. And of course, we can't talk about cultivated meat without talking about the path to market milestones. And the first really important milestone for the industry was in December 2020 with the first cultivated meat product that was approved and was available for commercialization in Singapore. That was the Just chicken nuggets. But then actually since then and quite recently, quite a few other milestones have been achieved. So last year, Upside Foods and Good Meat, they've received green light for safety for their products. And actually this year in around June time, no, not June, March time, around March time, both Good Meat and Upside actually received their regulatory approval for commercialization of these two products in US. And that is a huge, huge milestone. And I have, I mean, there has been media announcements as well that several other companies have submitted their portfolios for their products for regulatory approval in Europe, which again is a big milestone. But those haven't really, I mean, those are still at the start of this achievement. I do know for sure that actually UK has also received two regulatory approvals, one being Aleph Farms that has announced it, but there is another company which I have no idea who it is. So it is still to find out soon. So thinking about how cultivated meat could be produced, this is a very, very general production flow that basically starts with getting the cells from the animal, isolating them, creating your banks of cells, and then culturing these in the lab, initially in small scale flasks, but then translating those cultures into bioreactors. And then there is this idea of, is there a differentiation step required or not? I guess a lot of people have different opinions. My opinion is that you do have to carry out that differentiation step. And the reason for that is because if we go back to that definition of cultivated meat, which is to mimic as precisely as possible animal-derived meat. Well, animal-derived meat does contain differentiated muscle and fat. So from my point of view, I think the differentiation step is needed. And obviously after the differentiation step, you will have to go through a step of formulation, which can take different shapes and forms depending on what kind of cultivated meat product you're trying to produce. If you're aiming for a non-structured product, which is more like a minced meat-like products, cultivated meat products that you can then shape into things like sausages or burgers or meatballs, then it's enough to just generate the cell types that are required, meaning the muscle and the fat, and then those would be formulated in certain ratios to form that non-structured minced meat. If you're aiming for a structured type product, so more like a fillet or a steak or chicken leg or whatever that might be, there would have to be an additional step where essentially those cells that you have produced at scale, the muscle and the fat, those would have to be seeded onto a scaffold where essentially they would do their job and create that shape for the structured meat that you are aiming for. So this is, again, just kind of explaining this production flow in simpler steps. If it would be to go for the mimicking of the meat, then you would have your proliferation followed by differentiation and then formulation. You will see from the analysis that I have done of the two products that have been recently approved, that hasn't really been the case with all of them. So there are still debates on if certain steps could be jumped or not. So putting it very simply, in terms of how can we produce cultivated meat, I would say there are main four puzzle pieces. The cell lines, which you would need, the media formulation that you would require to grow those cells in, the bioreactors and the bioprocessing, and the scaffolds that can come in different shapes and forms. So in terms of the cells to start with would have to have the capacity to differentiate into the cell types that are component of meat. So that means ability to differentiate into muscle, ability to differentiate into fat. And actually, in a survey that was carried out last year by GFI, it turned up that the preferred cell types to start with would be myosatellite cells, which have the ability to differentiate into muscle cells, and fibroblasts, which is it was quite surprising. I mean, fibroblasts are fibroblasts. They do not have the capacity to differentiate. They're just connective cells that, yeah, indeed, they are found in meat in a smaller proportion, but I see them as essentially the glue that keeps all the other cell types together, but without having necessarily a specific function. To be honest, I was expecting more to see of mesenchymal stem cells as preferred cell types, because these have the ability to differentiate towards fat. And there have been some studies where they have been trans-differentiated into muscle as well. So so the results of the survey were somewhat surprising with the fibroblasts. So in terms of how many cells do we really need? Well, in a publication from Mark Post from 2020, he estimated that for a ton of cultured meat, you need to achieve somewhere around 10 to the 14 cells. And actually, in one of our recent book chapters that we've published, in the advances in cultivated meat production, we did a bit of we essentially looked at a few scenarios on how you would be able to achieve those sort of numbers, and what we concluded were that you would have to reach the scale of 10,000 liters, but at very high densities, cell densities. And I'm talking about 5 million to 10 million cells per mil cell densities. And then kind of looking at the timelines for differentiation. Muscle differentiation generally happens a lot faster. We're talking about 10 days at maximum, but fat differentiation or differentiation towards adipogenic lineage takes around 21 days. So actually there would be the need for a lot of batches in order to achieve those sorts of cell numbers. And I guess the takeaway message from this is that those sorts of cell densities that are required, they're not realistic, not yet anyway. Just to give you an idea, with mesenchymal stem cells that have the ability to differentiate towards fat, let's just say currently the cell densities that can be achieved, they are below 1 million cells per mil. So there's still a long way to go to reach 5 million and 10 million. With myosatellites, these type of cells, they would grow a bit faster. So realistically, at this point in time, they can be grown up to something like 2 million cells per mil, but that's still not close to what is required. So I think in terms of the challenges that still exist and are still to be addressed, getting those scales and getting these cell densities that are required, it still hasn't been solved. So there's still a lot of opportunity for development there. And another thing, just to kind of give you an idea of what is at the moment realistic and what is not, because not a lot of people talk about these, but I always like to show them what are the scale expectations depending on the product, or at least what has been achieved to date. So thinking about things like proteins or monoclonal antibodies that can be produced using recombinant DNA technology, those are at scales above 10,000 liters. So that is achievable. Products like viral vaccines, they are at around 2,000 liters, but products that use stem cells as the actual product, they are only at maximum 200 liters so far. And there are different reasons for this, right? I mean, there are one of the main reasons is obviously the fact that stem cells grow a lot slower compared to bacteria or yeast cells that you would use to produce monoclonal antibodies. There's also the issue of cost. The media for bacteria or for yeast cells is a lot cheaper than it is for growing stem cells. So in a way, I can understand why we haven't really progressed as much beyond 200 liters. But I think when thinking about cultivated meat and the type of scales that are required to produce tons of cells that would be the equivalent of tons of meat, we're not really there. I mean, we're nowhere near there. So these are still existing challenges that need to be addressed. It would be possible to reach these sort of scales using more robust cells. So maybe by immortalizing some of these cells, these challenges could be overcome, and these drawbacks when it comes to cultivating, to culturing stem cells could be overcome. Right, so thinking about those two products that have recently been approved, and again, as I said, this was based on their actual applications that they submitted to FDA, which are publicly available. So if you Google good meat FDA application, you should be able to find their actual applications, which I think is fantastic that FDA release these to the public. And there are really interesting reads in terms of how they assessed all the risks with these type of products, etc., and how they've written them without actually giving away too much or giving away proprietary information. There's very little that is actually redacted on those as well. So I was quite surprised from that point of view. But kind of thinking about those puzzle pieces that I said. So in terms of the cells, Both of the companies have taken a very, very different approach. So Good Meat, they've chosen to use an ATCC cell line that was established, I think, around 1980s. And this is an immortalized chicken fibroblast cell line. They say it was, this is based on data from ATCC, that the cell line was not engineered or genetically modified, but it was spontaneously immortalized and that it is genetically stable. ATCC did test this cell line to see if they form tumors, and they found that they don't. On the other side, Upside Foods, they've isolated their own chicken myosatellites and also fibroblast cells from animals. And then they actually adapted these cells initially to suspension cultures. So for those of you that are not familiar necessarily with cell culture, generally all the cell types that you develop from tissue, you isolate from tissue, they are what we call adherent-dependent cells. So that means that they need a surface to attach to. Without that surface to attach to, they don't survive unless you adapt them to the suspension culture. Suspension culture means they don't need that surface. They can freely float and they would grow, they would be happy. This type of behavior, so suspension of cells growing in suspension, generally is naturally characteristic to cells that come from blood or bone marrow or stem cells that can form blood cells. All other cell types are adherent naturally. But you can adapt them in culture to this sort of condition. And actually a lot of the cell lines like mammalian cell lines like CHO, for example, Chinese hamster ovaries, which is used on a regular basis to produce proteins, they have been adapted over time to suspension cultures. And from a manufacturing point of view, or from the point of view of a bioprocess engineer like myself, it's a loss. It simplifies the process if your cells can freely grow in suspension. Because if you have to use that attachment substrate, then obviously that's another layer of complexity. So Upside went down this route of isolating their own primary chicken myosatellite cells and fibroblast cells, adapting them to suspension culture, and then they actually spontaneously immortalize these cells. They say immortalized / bioengineered, so they have used something to get them there. So I wouldn't say is necessary. I mean, this kind of terminology to me is a little bit perhaps misleading because it's either one or the other generally. But yeah, they say it was somewhere in between, so I don't really know what that means. They did say that these cells were phenotypically stable as well. So that was basically to demonstrate the safety of these cells. In terms of the process, Good Meat, they said no scaffolds were used, which makes sense because their cells were immortalized and adapted to suspension culture as well, their fibroblasts. They don't have a differentiation step. And they did mention that the scale that they would grow, they would use to grow these fibroblasts would be 1,000 liters. They haven't built their own facilities, manufacturing facilities, but instead they've chosen to use a CDMO, which stands for Contract Manufacturing Facility. So someone that essentially that's their business model. They have the manufacturing facilities and they can rent them out or whatever, support the protocols of another company to produce them. On the other side, Upside Foods, it's quite an interesting approach that they've taken. So for their proliferation step, because the cells have been adapted to suspension cultures, they just grow them naturally in suspension. But actually for the differentiation step, because they do differentiate their myosatellites, they say that they are using a proprietary design for a bioreactor where essentially they adapted their cells back to attachment, and they detach the cells from the differentiation bioreactor as cell sheets, intact cell sheets. So I thought that was an interesting approach, because I guess what they're doing with the cell sheets is then they layer them up to achieve the thickness of their product. So by doing it this way, they can achieve the actual texture of their product. They did construct, build their own production facilities, but they haven't said anything about the scale that they're using, at least not for their bioreactors. In terms of the media formulations, Good Meat has gone down the route of using a fetal bovine serum in their media, so a serum-derived media. They say they're not using any antibiotics in their media, which is great, because that's one of the biggest selling points of cultivated meat. And actually, they have tested their final product after formulation and after doing the washes, etc., to see if there are any traces of serum left. And what they said was that it was 12 times lower than the serum that is naturally found in the milk that we find on the shelves of supermarkets. So that was deemed by FDA as a safe approach, even if they're using serum. Upside Foods, interestingly, their application is, oh yes, and Good Meat did say that actually they are developing their own serum-free media formulation now, but once they have that and once they've validated their process, they will submit an amendment to FDA for their serum-free media production. Upside Foods, they're actually taken this one step further, and they submitted for two processes, one with a serum-based media and one with their proprietary serum-free media formulation. So they've covered both of their bases essentially in doing this. Again, they said their media doesn't contain any antibiotics. They did admit that they have used antibiotics and antimycotics, but only in the cell isolation step, which, to be honest, you can't isolate cells without these. It just wouldn't be possible. So that's realistic. I think as long as antibiotics, antimycotics are not present afterwards, then I think that's fine. And then they also did an analysis to see how much serum was left, and they found the same thing, that after washes and after formulation, it was less than 12 times the amount that is found in the milk on the supermarket shelves. So in terms of media formulations, because media has been found to be the largest cost driver. Generally, media is formed from a basal medium, something like, and there are different versions of these, but essentially this contains the source of carbon, like glucose, source of nitrogen, which can be glutamine. It contains salts, vitamins, amino acids, buffers. So all the things that actually the cells need in order to grow. But this is not enough. So this is generally supplemented with the serum. And serum can be, depending on the types of cells that you're using, they can come in, it's needed in different ranges, different concentrations, generally between 10 and 20%. And serum is really good because it contains a lot of the other things that the cells need, like hormones, like insulin, proteins, where albumin is really important, transferrin is very important as well, lipids, growth factors, but then also trace elements like zinc and copper. And serum is a byproduct that is harvested from cattle. So some might argue that actually, you're not really wasting everything from the animal derived from the animal industry, but you're using some of the waste products to create other products of value, like serum, that can be used. And in terms of advantages, serum has been used on a regular basis, particularly in research labs, because it's so good at supporting cell growth. And in this context of making cultivated meat, is not necessarily, it doesn't necessarily come with a safety issue, in the sense that it comes from livestock most of the times, from bovine. And actually, if you think about the meat that we purchase from the stores, it has some serum in it because it has some blood in it, right? But the idea is that you don't eat it raw, you cook it, so that you bypass any of those issues. I think the disadvantages of using serum, particularly to make cultivated meat, comes more from the fact that it is quite expensive. And actually the price of serum over the past couple of years has doubled, if not tripled. And the reason for that is the availability of the animals. So that has been one of the main issues. There's also issues with the fact that the batch-to-batch, there's this batch-to-batch variability, which actually translates in differences in how the cells grow. So if you have a manufacturing process where you're using one batch of serum to grow your cells, the next batch might not give you the same results. So there might be significant differences. It is quite limited now in the availability. And again, this is linked to the animal availability. I've been to a conference this year in March that was organized by what is known as the International Industry Serum Association. So they're kind of a body that manages serum everywhere, so worldwide. And they were talking quite a bit about the fact that actually the regulations around the world, including Europe, have become a lot more stringent when it comes to serum. And that also includes the fact that now the kind of testing that they have to do on the serum that they sell to us is a lot more stringent. They have to do a lot more testing, or particularly on specific viruses. So obviously all those things will contribute to the rise in the price of serum. And then in the long term, there is a concern that this might not be sustainable. And again, because it comes from multiple animals, it comes from different parts of the world, the actual exact composition is not really known and is very, very dependent on its source, where it comes from. So there have been attempts, and actually quite a lot of companies are working towards trying to develop their own serum formulation. And even in research and academia, there's a lot of initiatives now trying to replace serum. And generally, the main approach is to try and replace all those components that I said serum has: the hormones, the growth factors, the lipids, the proteins. But when you look at the cost of all of those individual components, that actually ends up being a lot more expensive. So it's not doable for, at least not for cultivated meat production. So other attempts to replace serum have been looking at trying to use growth factors that can be produced specifically for this, so not as high purity as you would have them for therapeutics, for example, or producing them in strains that, bacterial strains or microbial strains, because you can also produce them in yeast, that are better adapted for, well, to be cultured cheaper, so that you can get cheaper growth factors. Another attempt to try and replace serum is taking inspiration from food science. So it's trying to use food grade components. And actually Will's PhD project is all about that. So he can tell you all about that if you want. And that can be a cheaper approach, but no one has really pinpointed it yet. So we'll see what will come out in the next couple of years. The same with using hydrolysates, and particularly hydrolysates from plants, plant sources. Using algae as well are options that can help replace some of these components, if not all of them. Other approaches are almost like a combination between looking at media development, but also combining that with bioprocessing strategies. So particularly looking at recycling elements. So you could have, as you can see here on the left-hand side, you can have certain parts of the media being recycled. And the idea behind this is the fact that generally we give ourselves more than they need. So a lot of products, a lot of those components that are found in media will be unused by the cells at the end of culture, and those just get discarded. So those are just waste. But actually by including one of these recycling loops, you could potentially bypass some of that waste, minimize some of that waste, and reuse some of those components. Another approach is to do co-cultures with other cell types. And this was an example that I've taken. I forgot to put the publication here, but it was a schematic that I found in one of the recent papers where they were using microalgae culture. And they were basically taking advantage of the fact that microalgae can utilize some of the components from the spent media, so some of the waste products from cell culture, and produce things that are useful and are nutrients for cell culture. So it could be almost like the circular type approach. And actually, thinking about the circular type approach and circular economy approaches, this is what the CARMA, or the Cellular Agriculture Manufacturing Hub, is all about. So it takes knowledge and opportunities from tissue engineering and precision fermentation and tries to combine those. So essentially is doing this by taking advantage of properties of certain microorganisms. Particularly what we have in mind is yeast, a specific strain of yeast that is really good at metabolizing ammonia. Ammonia is a waste product of animal cell culture, but it's actually quite detrimental to cell growth if it accumulates in culture. So by using it and feeding the yeast can actually produce other things that are useful for mammalian cells. So there's this, this kind of circuit of, or this idea that by collecting all that spent medium, all that waste medium from tissue engineering applications, so from cell culture, and utilizing it as a feed to grow yeast, and then vice versa, the actual waste product from the yeast culture as a feed for our mammalian cell culture, you can actually minimize the cost, you can minimize the waste, and this would be a more sustainable approach in the future. It's still early days. I mean, we haven't even started, to be honest, but because the project has only started in October. But I think the concept is definitely really cool, and is definitely something that you know, in a year or so, we'll have a better idea if it works or in what proportion it works. So the Cellular Agriculture Manufacturing Hub, CARMA, is actually a really big milestone for UK, in the sense that the government, so through the UKRI, the research councils, has invested quite a bit in the alternative protein space research now. So this was one of those investments. And the CARMA Hub is a £12 million, 6 years long project that is led by University of Bath and collaborators with UCL, University of Birmingham, Aberystwyth, and Royal Agricultural University. And it also has quite a lot of industrial collaborators, and I'm sure you probably recognize some of these logos. And these are just some of the initial ones, but I think the idea is that the CARMA Hub will act almost like a platform that unifies not only the academic research happening in the UK in this space, but also bringing together some of this industry and working together to address some of those challenges that I mentioned, like the cost, the sustainability aspect, and actually more than that, the CARMA also focuses on the training of the new talent, which it is quite widely recognized that actually there's a big gap there. So CARMA is all about skills, but with society, environment, and economy in mind. And these are just some of the main themes that CARMA will be addressing. So it's excuse me, it's not just about research into manufacturing, but it also has elements of understanding the public perception, looking at the regulatory aspect of the manufacturing process, doing life cycle assessments, and interacting with policymakers and informing some of those decisions when it comes to manufacturing alternative proteins. So I'll leave it at that. I have no idea how long I was talking for, but I'm happy to take any questions now. This is just the book that I just mentioned, that it was published recently and is called Advances in Cultured Meat Technology. So this was edited by Mark Post, Kate Conan, and Chris Bryant from University of Bath. So thank you very much for your attention, and happy to take any questions now. Any questions? Let me see if there are.
So you mentioned about how both outside foods and good use both immortalized cell lines.
Yeah.
And for quite some time, that's kind of a taboo word to kind of incorporate that in the bio process. But it seems funny that those two come first. And I'm wondering why you think that is. Is it cost or ease or reproducibility?
I think going down the immortalization route is obviously easier because you're bypassing those limitations of primary cells. These two companies, you do have to bear in mind that they are both in US. US is a lot more open to GMOs than Europe is, and UK is. So I think that sort of approach works well for US and probably works well for other parts of the world. Thinking of Singapore, I think they're a lot more relaxed, but I don't think it would necessarily work in UK or in Europe. It was kind of the same thing with Impossible Foods Burger, wasn't it? Because both Beyond and Impossible, they come from US, they're based in US. Beyond has made it to the market in Europe and UK, right? You can find it in shops like Tesco's, in supermarkets like Tesco's. But the Impossible Foods Burger, you can't find it outside US. And the reason for that is because they use a recombinantly modified component, the hem that they use to give the taste and the color. Yeah. Yeah. Okay. See. Oof. So how long cells in the meat will be viable? Can it be possible to make chicken wings in the future? Well, I hope so. I think chicken wings are probably, well, they're part of that structured meat category. So you would definitely need some sort of a scaffold to give it that shape. Um, but yeah, well, I mean, why not? There's a lot of companies that are going for structured products. How long the cells in the meat will be viable? Well, I think at the end of the day, you only need the cells to be viable while you're producing them, so you can get as many cells as possible. But then when you get to the formulation stage, you don't need them to be viable anymore, because at that stage, it's meat is the product that you're going to cook and eat anyway. I did read about some interesting approaches where, because there are debates that actually with meat, and particularly certain types of meat, to get that taste, it has to go through a curation or maturation step, but that maturation step happens in the presence of microorganisms from the environment. So I've read this study where they were trying to figure out if actually by getting the cells um, and then exposing them to the environment, if they would be able to achieve a similar effect of curing. So yeah, I thought that was an interesting approach. Is chicken the most popular cultivated meat choice for CELAC companies, and why? Yeah, that's actually a good question. I think it's only certain companies that are going for chicken. I think chicken is a lot easier to grow. And in a way, you can, I mean, logically, if you think about the lifespan of each of these species, right? Birds will leave, their lifespan is the shortest. So essentially, their cells will be the most actively growing, the most metabolically active, they will grow the fastest, etc. So while if you think about cows, they have a lifespan of what, 20 years. So their cells are naturally slower than the cells of chicken, right? So I think a lot of well, at least these companies, and I think there are a few more, have gone for chicken because of this reason, because it's probably easier to grow these cells. They're a lot more active. But there are, I mean, Moza Meat, which is the company of Mark Post, with the first burger, they're going for beef, and there's actually a lot of companies that are going for beef as well, pig. So I wouldn't say the majority prefer chicken. I think chicken just happened to be the first ones that came to market. But yeah, I think we are going to see more on beef, pork, and the other species as well. So has there been much work on achieving high cell density using perfusion bioreactors? Oh, that is a very good question. Not that I know of. I know there are groups that are interested in doing perfusion, and I mean, so when I was working many years ago, when I was doing my PhD and we were working with human mesenchymal stem cells, we have tried to adapt a stir-tank bioreactor to perfusion culture, and it has worked wonders. But it is quite a difficult thing to do in the sense that not all bioreactors have this capability of being adapted. So they need to have specific configurations in order to be able to adapt them to perfusion culture. And also with perfusion culture, there's the consideration of cost, because you are going through 3 times more the amount of media that you would go if you would do a fed batch system. So I think it's kind of finding that balance. But I do think perfusion could be a way to go to increase those cell densities. There is, however, limitation with using perfusion at scale. You can use it up to a certain scale, but not beyond. What would be the expected environmental impact of this new tech, such as processing waste, land use, transport, etc.? Yeah, I mean, that's a really good question. I don't think anyone really knows the answer to that. And I think why the environmental impact is not necessarily known at this moment in time is also because there can be so many routes they can go down to in terms of producing these products. I mean, you've seen Upside and Good Meat, they've gone completely opposite in pretty much all of those 4 pieces of puzzle, right? So there's so many variations of this manufacturing process that it's quite difficult to assess what the actual impact in the long term will be, at least now at this moment. I think that if this type of approach of recycling waste or revalorizing waste will be successful, I think that's what will drive a huge positive environmental impact for sure. But in terms of land use, I mean, if you don't need the land to grow the animals anymore, obviously you will save significantly on that. So land use is one that I think we can all definitely say that there will be significant saves on that. But in terms of the waste, transport, all of that, I know there are companies that are going down the concept of almost having like a self-sustainable farm where you have your animals, you have your whatever else you're growing. If you're identifying certain plants that you can hydrolyze and, for example, produce hydrolysates that you can use in your growth media, you could do that in your own farm. So then you don't have to worry about transporting raw materials from, who knows where. So yeah, there's a lot of imagination, I think, and a lot of interesting concepts out there. Did you raise your hand?
I didn't, but you
okay. Okay, you ask your question and then I'll read.
So, sounds like this is very receptive. Where do you think UCL's role in this ecosystem is, and what can you be doing more?
You mean in the CARMA or
in the
general? I mean, there's a lot that we can do. I just need the funding. Yeah, I mean, if I would have the funding available, or if there would be schemes, funding schemes to apply for, I think the next kind of things that I would focus on would be fish, cultivated fish, crustaceans. So I would really like to do some work in that space. Obviously still utilizing my skills and expertise in bioprocess development, because that's where I'm doing the best work. Yeah, I think I would really like to kind of expand on fish and crustaceans. I don't think there's really anyone else in the UK that is really looking at that yet. It could be because, it's sourcing the actual very fresh fish might be a big challenge. Yeah. And then obviously this work that is about to start with the CARMA on looking at waste valorization is definitely something that I think we can do a lot in that space. Yeah, it could be it would be quite interesting to see what comes out of it. Okay, so are foodborne diseases less prevalent in cultured meat? Theoretically, yeah, you shouldn't have any foodborne diseases. I mean, that's the whole purpose of the regulatory approval process. So actually, as one thing that I kind of learned from reading these FDA approved portfolios from Upside and Good Meat was the kind of things that the regulator would see as posing a safety risk. And that goes down to the cells that you're using. And actually, one thing that kind of surprised me, because I would have not naturally thought about it, was because even if both Upside and Good Meat used chicken cells, they still tested their product for bovine viruses and for porcine viruses. Does anyone have any idea why they would have done that? Why do you think? FPS. So they were using FPS in their culture, which is bovine derived. So that can be a source of bovine viruses being transmitted. So they were testing for that. And porcine, they were using.
Yeah, I mean, when you're doing husbandry, cattle, it really depends on the source. You don't know that.
No, so it wasn't because of that. It was because they were using porcine trypsin to harvest their cells. Trypsin is a proteolytic enzyme they use to detach the cells. And they were using porcine trypsin, so they had to test for porcine viruses. So I would say, in terms of foodborne diseases, I mean, the chance of getting those is very, very low to zero. Yeah. I wonder what can be done to speed up the reproduction rate of animal cells. Is there a limit? Considering the current cell division time in manufacturing processes, is it comparable to the in vivo rate growing rate? Oh, that's a good question. I think in terms of how the cells grow, one thing that I have found is that it does depend on several factors, right? So it depends on the age of the animal. It depends on, for example, thinking about cows, of bovines in particular. If you're getting cells from dairy cows versus cells from cows that are essentially grown for meat, right? There are differences in how their cells grow. Because they kind of have different functionalities, those cows. And actually all those functionalities translate to the cells, to the cell growth, cell potential. I've also found differences in breeds, cow breeds. So some breeds will generate cells which grow a lot faster or have a higher ability to differentiate, particularly towards muscle. So if you think about, if you do a bit of research into cow breeds, for example, you'll see that some of them are more muscular than others. And it's because some of them actually have a natural genetic mutation that allows them to grow a bit more. They're called, because I was doing a bit of research on that, and they're called double muscled breeds, even though apparently they actually don't grow twice the amount of muscle. It's only about 20-25% more. But I think it's because they look a bit more defined and less fat, etc. So leaner. And those breeds seem to give better muscle cells, but their fat cells, they're like not as good. Yeah. Oh, you mean from the animal? Yeah, I mean, generally how this is done at the moment is from animals that, you know, they've already undergone slaughter for meat. So you just get your tissue, your piece of meat, the tissue from that animal. Post-slaughter, we're talking half an hour, maximum an hour, and then you process that. I mean, that animal has already been committed to the food chain. I know there are some companies and some groups that were talking about isolating the cells from live animals and getting biopsies, but actually I think you're causing a lot more distress to that animal by doing it that way. Because you don't, you know, from a piece like this, which let's say it wouldn't cause much damage to the animal if it's a live animal, you're not really going to be able to get as many cells as you would need. So it wouldn't really be sufficient, and you would have to keep going back to the animal to be able to do that. And I think it's more harm caused in that sense. Yeah, you're not slaughtering the animal, but then you are causing it pain, right? So what's better? I don't know. Yeah. Okay, so yeah, I mean, to be honest, I don't know what the answer is if the cell division time is comparable to the in vivo rate. I would think it's similar, but I don't think anyone has actually looked at that, to be honest. Yeah. Is culturing structured or unstructured meat more complex, and which has more technological advancements? Why? I mean, they both have to go through the same processes up to, you know, the final step. So the unstructured, well, both the unstructured and the structured need cells at the end of the day, and you need loads of cells, right? So you will still have to go through that seed train and grow them in bioreactors, etc. It's just the difference with unstructured, you're just combine 90% muscle cells with 10%, let's say, fat cells, mush them together, and then you can process them in whatever you want, meatballs or whatever sausages. If you want the structured one, then you need that additional step. And actually for that, for achieving that structure, there have been different approaches. So either using a scaffold that gives it the shape, and you're essentially doing a co-culture of muscle and fat cells. Or there have been approaches like Aleph Farms, for example, from Israel, they're using 3D printing to create their steaks. There are questions, or let's say some concerns with the scalability of 3D printing, but also with achieving the thickness of their cuts. But yeah, other groups have gone down the approach like upside with their layering to achieve that thickness. So there are different options of achieving that. More technological advancements, I think, I mean, it depends what you're really looking at, because I think it's, there might be some advancements on that layering element or 3D printing, not quite sure, but I think there's opportunities for advancements everywhere anyway, in all stages. Yeah. Yeah, so usually that can be achieved. So the differentiation can be achieved either using what we call molecular stimuli, so molecular cues. So you're giving specific molecules that will signal the cells to go down to that differentiation pathway. Or there have been studies in tissue engineering where they used topographical cues. So for example, they used certain geometries of surfaces that again drove the cells to go down that route. But I think the easiest is to do it using molecular cues, because you're just supplementing the media with those substances to drive the differentiation. Yeah, so I mean, it's a good question. I don't know, it depends. So Upside Foods, they did it in a separate bioreactor. They had their own design. I think is doable in the same bioreactor, and actually by doing it in the same bioreactor, I think you're saving on costs because you don't need another, and actually you're kind of simplifying the process, the manufacturing process. But it's a bit trickier to do it in the same bioreactor. So I think with muscle in particular, there is the challenge of achieving another stimulus, like a mechanical stimulus, to drive the maturation of the muscle. Like think of, you know, fitness and bodybuilding, right? They use exercise as a means for that mechanical stimulus. So something similar should be included in the process to be able to drive that differentiation, essentially. Yeah. Okay, let's see what's next. Yeah.
I was just wondering if there was analysis out there, like comparing developing a more expensive media versus like the positive impact of the like environmental impact of having cultivated. Because to me, it's a little counterintuitive to using FPS in like a culture system.
Yeah, I mean, there have been a few studies, but because they were based on assumptions, because no one has really done this successfully. And even if they have, like Upside and Good Meat, you would think they already have processes. They're not really making that knowledge and information available. So some of these studies, they kind of ended up, because they were based on assumptions, they ended up being a bit contradictory. Some of them found that there's going to be a significantly positive impact. Others found that actually, it's going to be detrimental. So I think it's just difficult at this stage because it's still so early on. It's difficult to do that sort of analysis without actually having actual data on the bovine cells or pig cells. And actually, I think every single species will be different in that. Yeah.
So, thank you for looking thank you everyone for coming.
Thank you. Thank you for having me. Cool. We'll do it. Oh, there we go.
So that's the snaps at the front. If you go.
Close.
Yeah.
Yeah.